So the next speaker is my colleague Ed Boyden. Ed is at the MIT Media Lab, where he's the Eva Tan professor, also at the McGovern Institute of Biological Engineering and Brain and Cog Science. He's a professor of neurotechnology, head of the Synthetic Neurobiology Group, co-director of the MIT Center for Neurobiological Engineering, and among many other things, the co-inventor of optogenetics, getting the brain to light up when neurons fire. So Ed is making his mark in many ways, including making brains stretch so you can start to map out the connections with his expansion microscopy. Microscopy. Go ahead, Ed.
Thanks. All right. Well, thanks for organizing this exciting event, and it's really great to be here. I almost titled this talk "The Future of the Meaning of Life" because I've been thinking a lot about what got me into neuroscience in the first place. And all through my scientific life, starting as a student, I wanted to work on projects that were at the border of philosophy and science. And my first project was a chemistry project when I was an undergrad, working on the origins of life, which didn't work, or you would have heard about it. We were trying to create life from scratch, and that failed. And then when I came to MIT, I worked in quantum computing, which is also sort of the border of philosophy and weirdness and practicality, and that was a lot of fun, but also was very difficult. And so third time's a charm, I went into neuroscience next, and was really fascinated by the questions of, why do we want what we want, and do what we do, and think the way we think? Going back to when I first got interested in that border between philosophy and science, what's the nature of suffering and happiness? And so that was kind of my original motivation for getting into neuroscience. It led me to really wonder, what is a thought? What is a feeling? Can we really catch the brain in action and see what these things are? And if we could understand these things, would it really help us understand what it means to be human, to have a brain, and to understand how the brain generates the mind? So this is a long-term goal, of course, and it has to be broken down into many parts. Obviously, we need to build theories of how brain circuits generate thoughts and feelings. And of course, to get good theory, it helps to have good data. And to get good data, we had to build a lot of technology. And so right now is a really exciting time. And I'll end on maybe some thoughts of where we are at this cusp between the explosion of brain technology development and also the idea that maybe it's time to start applying these tools in concerted ways in order to understand how brain circuits are generating things like decisions or emotions. So it's helpful sometimes to take a step back and ask, why is this such a difficult problem? And I think there's two parts to the answer. So one is space. Brain cells are enormous. In our own head, they're centimeters in spatial extent. Going down a spinal cord, they're a meter in length, by far the largest cells in the human body. And yet, if you care about information processing, that's performed by very, very tiny compartments called synapses, which are where neurons communicate, and also compartments even within the cell that are made out of biomolecules, tens of thousands, maybe millions of different kinds of biomolecules. And so if you want to understand how brain circuits are computing, how it goes from sensation to action through decisions and emotions and memory and all that stuff in between, you really have to somehow cross all these different spatial scales. And that's a big challenge. And the other problem, in addition to space, is time. If you care about learning or memory or the acquisition of language or the progression of Alzheimer's disease, these are things that take place over years or even decades. And if you want to understand things that are really relevant to the human condition, then you have to somehow also not ignore the briefest events in the brain, these electrical pulses that last perhaps a millisecond in duration that are generated within brain cells, and the chemical exchanges that occur between brain cells at those synapses that I mentioned. Now, this is not just out of philosophical interest. There's also a lot of pressing medical need. And so on this slide are some of the diseases or conditions of the brain that I think many of us have heard about, or some of us, of course, have encountered in our own lives or with our loved ones. And that's also sort of very daunting. I mean, there's really no cure in full for any brain disease. There are treatments for some of them, but those treatments are, for the most part, partial and have a lot of side effects. So this brings an extra additional moral focus upon understanding the brain. One could argue that the complexity of these diseases is a direct consequence of the complexity of the brain that we just talked about, the spatial and temporal complexities, amongst other things. And so if you look at actually how long it takes to develop brain therapies, these numbers are actually out of date. I think the numbers are worse by now. It takes a long time to get a technology like a pharmacological agent out of the lab and into the marketplace, about a decade. I think it's up to 13 years, if I recall. The failure rate is very high. The cost is enormous. I think a study from Tufts University says it's more like $2 billion now per attempt. And as mentioned on the previous slide, very often the techniques are partial in effect and have side effects. So there's sort of two overarching sort of themes then that I think frame today's talk. So one is, can we really understand the brain to the point where we can make a biologically realistic, but ideally human understandable computational model of how a brain circuit is generating something interesting, a sensation or an action, a decision or an emotion? And also, can we understand brain disorders? Can we pinpoint how they begin? And maybe if we can't do that, can we pinpoint where to intervene in order to help the most? And so for the last decade or so, our group's been very focused on a couple areas, some of which are represented here. But one of the big directions has been to build 3 tool sets. Can we watch the brain in action? The brain cells are exhibiting these electrical pulses within them. Can we actually see them and eavesdrop on the actual ebb and flow of information during the computation of a interesting behavioral outcome? And then watching is nice, but can you control as well? Can you perturb? If you can perturb the electrical dynamics of the brain, then you can test in a causal way what is actually important. And furthermore, perhaps open up new understandings of how to interface to the brain. What actually does the brain want in terms of the inputs and expectations in order to make the brain do what you want to do, whether to heal the brain or to test out an idea of how a specific brain function originates. And then finally, we need the geometry of the brain, and not just the shapes of the cells, but where the molecules are. You know, if we could have a wiring diagram of the brain and also know where the key molecules that are exchanged, the transmitters, and where the receptors that listen to them are located, maybe we could stitch these data sets together. Imagine a neural network being modeled in a computer based upon these maps along with the molecules. And there are going to be a lot of unknown parameters, right? We're going to have a lot of clues about how the network is configured, but there'll be a lot of unknowns as well. So how do we guess those unknowns? Well, there's lots of ways of doing statistical inference and understanding how to fit parameters of functions. In the most simple domain, we have curve fitting, gradient descent, all these other strategies for inferring the parameters of a, let's say, set of coupled differential equations. And one possibility is that by observing and controlling those high-speed dynamics, you would be able to observe what happens in a way that would allow you to guess those functions' parameters. And this is something that actually many of us in this room have been talking about. And for example, Max Tegmark and several of us have been working on proposals of how to stitch these kinds of data sets together. Okay, so let's do 3 short stories about these 3 kinds of tools, and then let's end on thinking about how can we stitch them together. Is it possible that we can really understand what a decision is, or what a behavior is, or what an emotion is in some finite time scale? So part 1, can you watch the brain in action? So Erika Jung and Kirill Perkovich, who were both postdocs at the time that we began this project, wanted to make a molecule that would light up when a brain cell was active. And not just any kind of activity, the actual electrical activity, the voltage of the cells. Now, if you look in the natural world for such a molecule, you're not going to find anything. In fact, maybe it's bad to be broadcasting your thoughts if you're some prey-like critter out there in the wild. And so the basic idea was, if the natural world's not going to evolve the thing, what if we could do it in the laboratory? And so in collaboration with a whole bunch of people, we built a robot that can do directed evolution. Basically, take a molecule that you think could yield a glowing indicator of voltage. In this case, they're fluorescent indicators, so we shine one color of light, and we watch a second color of light that's emitted. Make lots of mutations, the way evolution works in the wild, and hope that some of them will be better at what you want, in this case, a glowing indicator of electrical voltage. Put the mutants in cells, mammalian cells in this case. And then basically what we have at the bottom row of the schematic is a robotic scientist. Can we watch those cells and look for the ones that have bright, glowing indicators of when electrical changes occur, which we can stimulate with wires? And then we bring in a robotic arm, suck out the cells, and therefore the mutants that are interesting, and then we can actually go and sequence them. So we started with a molecule that was developed by Adam Cohen's group at Harvard, which was a weakly fluorescent indicator of voltage. And we made zillions of mutants. And in the end, we're able to create a molecule that actually does have this effect. So here's some work that is still kind of unpublished, still kind of under review, collaborating with the Han Lab at BU. And what you can see here in the magenta traces are what look like electrode traces. If you've ever seen wires that are put into the brain and you record the electrical pulses that they create. But these are not being recorded by an electrode. These are being recorded by a camera. And you can see a schematic in the upper left of what this looks like. You can shine light from a red laser, and then there's a fluorescence filter that collects the redder light and sends it to a camera. And now we can monitor neural activity without having to jam a wire into the brain. And so one of the interesting questions now that we're working on in the lab is how big a population can you see? Why can't you see hundreds, thousands, maybe even more of brain cells? And so in the coming years, it'll be very interesting to see how big one can scale up the ability to monitor these electrical pulses in brain circuits. Now, watching is great, but we also would like to be able to perturb. And so many years ago, my co-inventor, Carl Dreyfoos, and I were brainstorming about how we could use energy to control the electrical pulses in brain cells. And we just started going through all the laws of physics. There's light, there's sound, there's mechanical force, there's radio frequency waves. And so we thought light would be great if you could pull it off, because light can be aimed at things, it's very focal. And also light is as fast as anything ever gets, so we could keep up with the comparatively low speed of brain activity relative to the speed of light. How do you do it? Well, you effectively need a solar panel, right? Something that converts light into electrical signals, and then install the solar panel in the brain cell. Now, this is a hard thing to do, but if you could do it, then you could potentially bring light in using an optical fiber or other source, or even if the color of the light is correct, beam the light in from the outside and non-invasively activate or shut down neural activity. So if you could causally test out what a pattern of activity means, you could try to see, is it sufficient to trigger a behavior or a change that's of interest? So we lucked out. It turns out the natural world has evolved, in this case, a molecule that can do this kind of thing. So this is a single-celled green alga that live in ponds that are full of pond scum all over the world. And that brownish spot in the back is an eye spot that has molecules in it that can convert light into electrical signals. So if we zoom into the eye spot, you'll see these proteins. This is lucky part number one, is that the molecule is a protein, so that there's a good chance we can install it into a brain cell. When it's hit by light, blue light in particular, it opens a pore and lets charged particles, ions, across the membrane from one side to the other. In the natural world, the algae uses this signal to steer the little flagellae and help it navigate around in the water so that it can photosynthesize optimally. Lucky part number two, the protein turns out to be encoded by a small gene. And from all the tricks of gene therapy, we can take that gene and transplant it into brain cells. Now, it doesn't guarantee that the gene will actually make anything useful. Maybe the gene forms something toxic. Maybe the gene product requires some chemical cofactor that has to be administered externally. But we again got really lucky. It turns out that you put the gene into a brain cell, and the brain cell will actually manufacture the protein. And it isn't toxic. And you can install the protein in the right place because somehow it has the right trafficking machinery to end up on the boundary or the membrane of the brain cell, which is where the voltage is. And even more serendipitously, if you shine light on the brain cell, those pores will open, no chemical cofactors or other machinery needed, and you'll get electrical pulses, not unlike the ones that are happening right now in your auditory system as I say these very words. So that was very serendipitous. So what can you do with this? Well, there's all sorts of tricks from the field of gene therapy, as I mentioned earlier, where you can target the expression of this protein just to some kinds of cells and not others. And so, for example, the brain contains so many kinds of cells, some small, some big, some excitatory, some inhibitory. But just as an example, there are cells that are very interesting in the brain, such as these little star-shaped cells that are known as basket cells. And they're of great interest in biology because they do change in many kinds of brain diseases. And groups like Li Wei Cai's group here at MIT have been perturbing them and trying to study the kinds of effects that they can induce. So in fact, her group, with our help, co-led by Hannah Iaccarino and Annabelle Singer, working across the two groups, they made a cool discovery. They work amongst, on other things, Alzheimer's disease, and they've used this technology that I just described, which we call optogenetics, opto for light, and genetics because it's a gene-encoded construct, and they drove the brain at what is sort of a magical frequency in neuroscience, 40 times a second. And if you do that, they found, surprisingly to all of us, that this actually can help in Alzheimer's model mice. These are mice genetically engineered to have some of the same kinds of genetic changes that happen in people with Alzheimer's. They're not perfect models, not by a long stretch, but they're a good place to start. And amazingly, when you drive the brain at this frequency, the brain's immune system turns on, and you can clean up things like amyloid plaques in the brain. So they made this pretty stunning discovery, but it gets better. The teams went on, collaborating with, amongst other people, Emery Brown, also here at MIT, and tried to figure out, could you replicate this effect without the optogenetics, without the genes, without having to beam light directly into the brain? And so they found that if you actually deliver light through the eyes, just watching a movie more or less, or a flickering light, that was also enough to entrain the brain and cause this effect. So we actually now have co-founded a company, Cognito Therapeutics, which is trying to effectively do trials of movies, more or less, to treat Alzheimer's disease. There are all sorts of other things people are doing with this technology. It's in widespread use. This is work from Chris Fiorilla's group, which I picked because it has a cool movie, where they are looking at dopamine neurons. I think many of us have heard of dopamine. In the popular press, it's sometimes called the pleasure center of the brain. What they did was to engineer mice using some genetic tricks so that you could have those cells, the dopamine-producing cells, activated by light. And their question was, what does activating these cells do? So they had a very simple behavior paradigm. When mice go to one point in a box, they get a pulse of light. And when they go to another point in the box on the left, they don't. And here you can see the outcome. The mouse goes, pokes its nose, gets a pulse of light, does it again. And so basically, the mouse is working for light. This shows, at least in the constraints of this behavioral paradigm, that activating these cells is enough to serve as a reinforcer and make the brain do more of what it was doing. Let's see how this works. OK. So I think the theme that's emerging is that, indeed, tools can be a big multiplier for the productivity of the biomedical world. And if we're talking about the future of the brain, then one of the things that's very interesting to think about is whether tools that we're developing now indeed are having interesting and nonlinear effects in the productivity of science. So the quest has gone on. We've been searching all over the tree of life for molecules that can have interesting properties. Another example are neural inactivators. We can turn neurons off. And this slide just summarizes 3 major classes of these molecules that have been very helpful in neuroscience and now that are in use by literally thousands, I think, at this point, of research groups. Why would you want to turn neurons off? Well, there are many cases where you would like to delete neurons and figure out what they're needed for. If you can delete neurons momentarily, and it has to be done very carefully with lots of controls, then you can silence them and figure out what they're necessary for. Or you can try to shut down a pathological set of neural activities. Imagine an epileptic seizure. Could you turn it off? And now, at least in mouse models, people are trying to shut down epileptic seizures or other kinds of pathological overactivities of brain cells. A fun example is from this study by Akihiro Yamanaka, who did one of the earliest neural silencing studies that was related to a specific disease. He was working on narcolepsy, and in narcolepsy, there's a certain set of cells that dies off, and now known to be most likely due to autoimmune changes. And you can put a light-activated inhibitory molecule, which is from a single-cell microbe, into the set of cells that make a peptide called orexin, or hypocretin, that's deep in the brain. And then you can shine light from a laser, and these circles are indicating the percentage of the mice that are awake. And you can see when the orange bar begins, the mice pass out, and they're all there at 0% awake. And then when the light turns off, they wake back up. So to summarize, we now have ways that we can watch neural activity in action, and we're hoping to improve the technology even further. And we have a fairly maturing set of tools now, which allow us to manipulate neural activity. So those are 2 legs of the tripod that I outlined at the beginning. What about seeing a map of the brain? So this is a difficult problem. How do you see a map of the brain? I think we've all seen pictures like this. This is too crude to see the fine wiring of the brain with an MRI scanner. At the other extreme, you have microscopes, which can see very small things, but not small enough, because light has a finite size or wavelength, and it's hard to see things that are much smaller than that. You can do it, but it's extremely expensive and slow. So in our group, we started taking inspiration from the work of an MIT physicist, Toyuchi Tanaka, who worked out the physics of swellable polymers in the 1980s and 1990s. And if you take the polymer sodium polyacrylate, which is like the active ingredient in baby diapers, and add water, it'll swell. And so in our group, starting with 2 then grad students, Fei Chen and Paul Tilburg, and now we actually have probably about half our group working on applying or optimizing or refining or changing the technology, we decided, what if you could weave a mesh of the swellable polymer throughout a piece of the brain and make the polymer so dense and so even and soften the tissue using chemical processing so that when you add water, the baby diaper polymer would expand and you'd expand the brain along with it. So in panel B is a piece of the mouse brain before we expanded it, and in panel C is the same piece of mouse brain tissue, I guess like a day or two later, and it's been blown up by about 100 times in volume, about 4.5 times in each direction. So the hope here is that we could take a brain cell, like the one on the left, and if you expand it evenly enough to make it like the cell on the right, where the biomolecules have all been pulled apart from each other, and like stars in a constellation in the sky, they're all hovering in space, but the relative organization has been preserved. These are just a few short movies explaining, for those who want technical detail, how it works. So in brown are the proteins, and we build anchors that actually can bind to them and give little handles to the proteins and other biomolecules so we can apply force to them. The anchors are shown in purple here. And then we have to form the polymer mesh. We can do that using classical free radical polymerization, where the monomers, shown as little white spheres, will self-assemble into these long chains. And when the polymer chains encounter the handles, they will form a covalent bond. So if you think about it, the polymers can make force, the handles convey the force. We have all the ingredients you need, except for one, which is that we have to soften the brain because it doesn't like being expanded. And we can use enzymes, detergents, heat to do that, so that when we add water, just as Tanaka worked out the physics of many decades ago, the polymers will swell, but this time the biomolecules will come along for the ride. So it's not perfectly even, but it's accurate within a few percent over length scales of tens to hundreds of microns. And so already we've transferred the technology to hundreds of groups where it's being applied to all sorts of things, where you want to visualize nanoscale things across extended lengths. Here's a movie made by several people in the group working together of a piece of brain tissue that's been polymerized, and then we added water right there. And I hope you can see it's growing right before your very eyes. Importantly, those polymers are very dense. Work from other groups, not from us, suggests that the spacing between polymer threads is very, very tiny, as low as a few nanometers. So what can you do with it? Well, there are things you can do, like color code neurons. We did not invent that. This is developed by people like Da and Kai and Jeff Lichtman and others, the rainbow technique. Basically, you put a fluorescent protein in a brain cell, it'll light up. And it makes beautiful pictures, but if you look at the top middle, it's hard to see exactly what's going on when we hit the resolution limit of the microscope. But in the upper right is the same image as in the upper middle, but it's been expanded. Of course, we shrank it down to fit on the slide. It should be a much bigger picture. But I hope you can see that the fuzziness of the middle top has become clear, and we can even distinguish individual wires in the upper right image. And so this is very exciting now. What if we could indeed make maps of the brain using combinations of color codes and the expansion process, and make very, very detailed maps of how neural circuits are organized? This is sort of a tangent, and I'm also out of time, so maybe I'll gloss over the next two things. But one of the things that's nice about working on the brain is there's a lot of other biological problems that share sort of intellectual structure with problems in neuroscience. So we talked about the difficulty of crossing spatial dimensions and temporal dimensions. There are other large objects like tumors that also you would like to understand at a nanoscale. And so when Yangshin Zhao was in the group, we worked with Octavio Bekur and Andy Beck and Astrid Wyans and others, and looked at breast cancer specimens. Could you expand a cancer and diagnose it more accurately earlier? And this is sort of the data at the bottom shows what happened. Basically, on the y-axis is the accuracy of a machine learning algorithm that was trained to discriminate between different tumors. And in the purple squares are when the training was done with unexpanded data, and in the green circles are when the training was done with expansion. And you can see that it did a lot better when the expansion process occurred. And we actually spun out a company, Expansion Technologies, to try to see if we can do early cancer detection. Another tangent is very often in our group, we try to think about doing the opposite of what other people are doing, or even doing the opposite of ourselves. And Dan Oran and Samardrikas and others in the group, we decided, what if we run the brain expansion process in reverse? So start with a pure baby diaper material, sodium polyacrylate, add water to swallow it up. No biology, by the way, no brain or anything. But you can then laser print interesting things like metal or semiconductors into the swollen polymer, and then dehydrate it and shrink it down. So this is sort of an interesting thing to do. And what we found is that we might be able to use this to make nanotechnology cheaply. So these are examples here where we pattern metal in panel D over here into the swollen sodium polyacrylate hydrogel. And then panel F is what happens when you shrink it down. And just as the expansion process is even, so is the shrinkage process. And so one hope here is that there are spin-off applications of these kinds of ways of thinking in making nanotechnology. OK, last detour, because we're almost out of time. OK, so closing the loop here. We put forth the hypothesis at the end of the talk that maybe if you made very detailed maps of brain circuits, after having watched and controlled their electrical activity, maybe one could actually try to synthesize computational models of how those brain circuits work. And then could you actually understand the processes that occur that are behind things like decisions or emotions and so forth. And I think that what we're seeing now is that the tool sets are getting to be quite not only useful but compatible, and can we integrate these tool sets in an interesting way. So going forward, one of the hopes here is to start a roadmap, starting with small brains but rapidly growing. And so right now in the group, we have work that's going on with small organisms like C. elegans and zebrafish, as well as larger organisms like mice. Human brains, of course, are enormous, but it's clear that if we can derive principles of neural circuit function by studying these faster to analyze brains, and then maybe also derive principles of how to scale that technology to larger brains, it could be feasible, potentially, to start combining these tool sets in an integrative way and understand not just how brain circuits work, but maybe over the coming decade or two, where to go after brain diseases as well. So I think I'll end there and take questions.
Thank you, Ed. There are a bunch of questions that we can ask, and certainly in the panel we can drill down a little bit more about it. But bringing it a little bit back to human augmentation, when you and I would first talk maybe 3, 4, 5 years ago, one of your projects was the brain coprocessor. Mm-hmm. Is that still in the books, or what exactly, how do you envision that to roll out? What do you envision that to be?
Yeah. So thousands of studies have been done with brain stimulation in humans through methods like transcranial magnetic stimulation. I think part of the problem is, since we don't have an underlying understanding of brain processing, the number of applications that have actually gotten out of the lab and into human use has been fairly small. There is an FDA approval in 2008 for transcranial magnetic stimulation for depression. But because of our understanding of the underlying processing is so poor, it's very hard to know in a predictable way how to generate new brain stimulation protocols. Okay. It's like we have a keyboard, but we don't understand what's going on in the computer. And so it's very hard to write a computer program. So in our group, I think one of the key insights is that if we could understand the computer itself, then maybe we could be able to interface to it better. Mm-hmm. And so we do have some interfacing projects that I didn't have time to talk about, sure, including a non-invasive brain stimulation method. But the biggest question is often, how do you use it? What's it good for?
Yeah, yeah. Do you really think that optical stimulation of the brain is a competitor to wiring it? Especially, what would you do with the skull? Are we going to have transparent skulls when we go into this realm, or is it more like what Mary Lou Jepson wants to do by inverting the diffusion with light going through the skull?
Yeah. So I kind of glossed over it because it's a short talk, but I think this idea of using these tools to make scientific discoveries, and then once the scientific discoveries are clear, is there a practical, maybe even non-invasive way to augment brain function, is a faster road. I mean, these are gene therapies involving genes from algae and other critters. How do we know that the body won't reject it with an autoimmune change or something? I really like the example from the story of Leeway, where optogenetics yielded a pattern of activity, but the way that we're now trying to induce the pattern in humans is through non-invasive means, through our eyes and through our ears. And so an interesting question is, could we design basically a compiler? Here's the goal of what we want to actually create in the brain. Can we translate it back out into something that we can create externally? In the same way that when you program a computer, it's eventually all zeros and ones, but most people like to program it in some language that looks more like human language.
Good. Well, let's thank Ed. I think we're about ready to start our panel.